All right. This is another short and precise video. This time around, we are taking it to the biology aspect. Of course, you're welcome to my YouTube channel. What we do here is just to give a quick breakdown, but this particular video is basically for those that are writing the UTME examination. Just want to give a tidbit on the 2025 UTME questions and then things that you should look at, some basic points very quickly. Now, there's a question here on the board. Just want to review some few questions. Please, as you watch the video, do well to share and do well to comment, and also recommend the channel to your friends that are writing examination. Yeah, so you may be watching this video maybe an hour to your exam, or 2 hours to your exam, or a day to your exam, but just pay attention to the basic things I want to review with this video. Now, look at this question. Which of the following animals are triploblastic? First of all, for an animal to be regarded as being triploblastic, that means he has three body layers. Now, what are the major three body layers? We have the ectoderm, the mesoderm, and the endoderm. Now, there are some organisms that have two body layers. There are some that don't have body layers at all, or body cavity at all. Now, for example, the coelentrates. When we talk about the coelentrates, I'm talking about the organisms like the hydra. We are talking about the jellyfish. We are talking about the sea anemone. We are talking about the Aurelias. Now, all these guys are regarded as what? As coelentrates. Now, these coelentrates have two body layers. And what are the two body layers? They have um, ectoderm and endoderm. They don't have the mesoderm. The mesoderm, instead of the mesoderm, they have a non-cellular membrane there known as the mesogly. So those organisms that have two body layers are said to be what? Are said to be diploblastic. Now, but they are asking us, which of the following is triploblastic? Of course, you can see Hydra. Hydra is out of it already because Hydra is a coelentrate. Hydra is a diploblastic what? Organisms. Now, remember, diploblastic organisms are also regarded as cnidarians. So cnidarians are diploblastic. That means coelentrate as cnidarians. Now, tapeworm is a platyhelminth. It's a flatworm. Now, it's triploblastic. Now, of course, we have another one, the corals. The corals also falls under the coelentrate, which are diploblastic. So you can see what we are brought at now. But talking about tapeworm, remember, tapeworm, sponges. Oh, we didn't say anything about the sponges. Sponges are poriferous, obviously. They don't have body cavity, so to say. Now, these tapeworm, they are parasites, not just parasites, they are endoparasite. That means it stays inside, they affect. But this tapeworm, there's some things that you must know about tapeworm. Yes, they are flatworms, they are platyhelminthes, and you must also know again that the excretory structure of flatworm is the flame cell. The excretory structure of annelids, of course, you should know what are annelids. Annelids are segmented worms. Talking about the annelids, we're talking about the earthworm, talking about the leeches. They are common examples of annelids. Now, other questions from tapeworm that they can ask in the exam is that, of course, the head of tapeworm is called scolex, and he has a body part which is proglottids. And again, you should know that immature tapeworm is bladder worm or cysticerca. Either you call it bladder worm or you say cysticerca. So the immature tapeworm is bladder worm or cysticerca. Now, there are two common species of tapeworm. One is found in pig, one is found in cattle. The one found in pig is Taenia solium, which have hooks and suckers, while the one found in cow is Taenia saginata. It does not have what? Hooks. Now, look at this tapeworm. We said just now it's an endoparasite. Now, looking at number 2 question, which of the following is the excretory structure of insect? Now, this particular question actually came out the first day of UTME examination that they wrote, the first set of questions that they brought out. Those first set that wrote saw this question. Now, the excretory, what performs the role of respiratory structure in insect is the trachea. So the trachea plays the role of lungs. Like in humans now, it's lungs, but for insects, it's what is trachea. Now, if they begin to ask you for respiratory organ, that means they may likely ask also for excretory structure. So if they say, what's the excretory structure for insect, for example, you should know is the Mammalian tubu. The Mammalian tubu is the excretory structure for insect. But when they say what's the respiratory structure, you're talking about the what? The trachea. So for insect, respiratory is trachea, but for excretory is the Mammalian tubu. Now, please pay attention to this. But remember, there's one common question really asked again, talking about that of spider. Spider is not an insect, obviously. So if they not say what is the respiratory structure of spider, is a long book. So for spider, it's long book. But arachnids, spider is an arachnid, and arachnids have 4 pairs of legs. Insects have 3 pairs of legs. So I just want to use these questions to remind you of some certain things that you should put at the back of your mind. So as you're preparing for the exam, know about respiratory structure, know about excretory structure. Now this particular question actually came out the first day, like I said just now, that they started the UTME examination. Now you have known the respiratory structure of insects, you know the excretory structure of insects. You know that spider is not an insect, spider is an arachnid. Spider uses long book as a respiratory structure. What about the excretory structure of spider? It also uses Mammalian tubu, just like the insect. Now, talking about spider, let's go to some next question. They said, which of the following is not an endoparasite? Of course, I made mention of endoparasite just now. Endoparasite, they stay inside, ecto outside. Ascaris is an endoparasite. Plasmodium, that causes malaria, is a parasite, of course, a protozoa. It's an endoparasite. It feeds on the red blood cells. Flukeworm, talking about the liver flukes and the rest, they are endoparasite. Now, lice is not an endoparasite, it's an ectoparasite. So endoparasite, ectoparasite, you must know. Tapeworm is an endoparasite, like we stated just now. Now look at this. This question is coming from ecology, one important aspect that you should pay attention to again. Talking about the biotic factors, the abiotic factors, terrestrial environment, factors that affect aquatic and terrestrial environment, factors that affect only aquatic, for example, salinity, turbidity affects only aquatic environment. For example, edaphic factor affect only what? Terrestrial. All right, now this is another question again that you must look at, considering the examination. Now they said this one is based on test anyway. That means we must look at how to test for the class of foods, talking about the major ones, the carbohydrates, the proteins, and of course the fats. But let's take this question first, so begin to elaborate on these tests. Now they say if Benedict solution gives a positive test or positive result in a food test, the food contains sugar, protein, oil, fat, or cellulose. Now first off, sugar, Benedict solution or Benedict test used test for sugars. Now it's not only Benedict that can be used to test for sugars. Saline solutions are also used to test for sugars. Because saline solution is a blue solution. When you use to test for sugar, it turns to brick red. So look at it. Thalins and Benedict solutions are used to test for what? For sugars. Now to test for starch, of course it's not on the ocean, but to test for starch you have to use iodine. Iodine is used to test for what? For starch. Now look at it. Protein, now there are various tests to test for protein. Talking about the Burette test, talking about the Millions test, the Xanthoproteic test. These tests are used to test for what? Proteins. Now talking about oil, let me summarize this together. Oil and fat, that's lipids. To test for lipids you use osmic acid. It can be used to test for lipids, and of course Sudan 3 test can be used to test for lipids also. Now of course, the polysaccharide, the cellulose, of course, you can hydrolyze it, then use what's it called, the Benedict test to test for it also. Now coming back, all the following are digestive enzymes except bile. Let me take the options: bile, lipase, maltase, pepsin, and thealline. Now coming back to this, remember, food digestion starts from the mouth, and there's an enzyme in the saliva known as thealline, which is also called salivary amylase. Now these salivary amylase, which is thealline, helps to break down starch to maltose. So this guy is an enzyme here. Pepsin, of course, is an enzyme that is found in the gastric juice, which is in the stomach. Of course, remember that when gastric juice acts on food, the food is now called chyme. Inactive pepsin is what is called pepsinogen, and it's HCl that usually helps to activate the gastric juice in the stomach. So yes, pepsin is an enzyme, breaks down protein to peptones. Now talking about maltase, maltase is also another enzyme that breaks down maltose, an enzyme. Lipase are enzymes that breaks down what, fats to fatty acid and glycerol. No, bile is not an enzyme, but bile is produced in the liver but stored in the gallbladder, and bile is involved in the emulsification of what, of fat, and also creates an alkaline environment for the action of intestine of pancreatic what juice. Now be very careful of these things that I just made mention of. Now that means you have to look at the digestive system, and of course this question coming out from test, you have to know the test, and of course go to flowers and have to check those questions on flowers. Now this is just a quick review that you can put at the back of your mind as you go into the examination. So I wish you all good success. So you can share these videos with your friend. Do well to subscribe also as you watch. Thank you very much. Much